Hello, I'm Manfred Meyer, researcher at the Chair of Plant Breeding at the Technical University of Munich. I would like to give you an overview of the phenotyping experiments that we were carrying out at the plant phenotyping platform PhenoArch, located in Montpellier in southern France and belonging to the French National Institute of Agricultural Research. The whole experiment has been made possible through an EPPN2020 Transnational Access Grant. The main goal of our research is to make the native diversity of genetic resources, such as landraces, amenable to crop improvement. Today's elite germplasm in maize shows only limited genetic diversity compared to the whole species. Therefore, for securing further breeding progress under changing environmental conditions, an introgression of favorable alleles from landraces is needed. In previous work, we generated a unique resource of 1000 double haploid lines derived from 3 pre-selected European maize landraces. We conducted extensive field trials in up to 11 environments for evaluating their line per se and test cross performance. Our special interest was on traits related to early plant development and cold tolerance. These are complex traits and generally hard to measure in the field. That's why we used early vigor scores and early plant height measures as approximations. The results of the field evaluations showed that the different measures of early development target different components of the trait, and their correlation with final plant height and biomass yield varied in magnitude. We therefore were interested in dissecting the complex trait early plant development further into its components and elucidate the underlying genetic basis and molecular mechanisms. For doing this, high-resolution phenotypes were required that only could be obtained in a phenotyping platform such as PhenoArch. With more than 400 DH lines and 100 test crosses, and with 3 replicates per entry, more than 1,500 plants were evaluated in the platform. Each plant is on its own cart located on a conveyor belt and can move around freely to watering stations, stations for manual phenotyping and maintenance, and most importantly, it daily goes through an automated RGB imaging system from which important traits related to plant growth, plant architecture, and radiation use efficiency can be extracted and monitored over time. The experiment revealed large genetic variation for the different components of early plant development in the analyzed material. Part of the landrace-derived test crosses outperformed commercial hybrids, which were included as checks in the experiment for early biomass accumulation. The experiment was successful in generating high-quality data that provide an ideal basis for upcoming analysis, like identifying novel beneficial variation for early plant development in landraces, advancing genome-based prediction models for vegetative development traits, and determining the relevance of individual trait components for increasing final biomass yield. The experiment took place during the COVID-19 pandemic, which unfortunately made a visit of myself to the platform impossible. I therefore want to thank the whole team in Montpellier for their support and full commitment in carrying out all practical steps of the experiment, which we discussed in online meetings on an almost daily basis. Thank you for watching, and thanks to EPPN 2020 and all team members involved in this project.